Hello everyone, welcome to Concept and Fact of Life Sciences. Today in this video, we are going to discuss about the plastids. As we already discussed in our previous video that the plant cell, they have three double membranous cell organelles which includes the nucleus, mitochondria and the plastids. The plastids, they are mainly of three types which are chloroplast, chromoplast and leucoplast, and this classification, it is based on the type of pigments these plastids have. For example, if we talk about the chloroplast, it is green in color, and the green color, it is due to the presence of chlorophyll pigments. And if we talk about the chromoplast, these are colorful plastids, and the color of these plastids are due to the presence of pigments which are known as the carotenoids. Now these carotenoids, they are of two types. One is called as the xanthophylls, while the other is called as the carotenes. So there are two types of carotenoids which provides unique colors to these chromoplasts. Now, the leucoplasts, they do not have any kind of pigments, so they are colorless. Now, these plastids, they also have different functions. For example, if you talk about the chloroplasts, they are mainly involved in the process of photosynthesis. If we talk about the chromoplast, since they provide unique color to the plant, so they helps the plant to attract the pollinators. So they help in the process of pollinations and also in the seed dispersal. Now the leucoplasts, they are mainly of two types, etioplast and amyloplast. Etioplast, the leucoplast which stores the fat, while the amyloplast are the leucoplast which stores the starch. Now these amyloplasts, they also act as a statoliths. These molecules, these amyloplasts, they help the plant in the process of gravitic perception. मीन्स ये जो मॉलिक्यूल्स हैं ये प्लांट्स को ग्रेविटी को सेंस करने में हेल्प करते हैं. कैसे बात करते हैं थोड़ी. Since we have taken a plant, the above ground part of the plant, it is called as shoot, while the below ground part, it is called as the root. Now these statoliths or the amyloplasts, these are the denser molecules which binds to the plant root in the direction of gravity, and after binding of these statoliths, there is a growth of root towards the gravity because these are the denser molecules. So they help the plant to sense the gravity. Now all of these plastids, they have common origin, that is from the proplastid. Now the proplastid, it is a undifferentiated plastid. Now the conversion of this proplastid into the different type of plastids like chloroplast, chromoplast and the leucoplast, it depends upon some external factors like the light, as well as on some internal factor, for example the plant hormone, that is cytokinin. So the conversion of proplastid into the different type of plastid, it depends on some of the external factor and also internal factors also. So the most important type of plastid here, it is the chloroplast, which is mainly involved in the process of photosynthesis. So let's talk about the chloroplast. The chloroplast, it is a double membranous cell organelle. ये एक डबल मेम्ब्रेनस सेल ऑर्गेनल है जिसमें दो मेम्ब्रेन्स होती हैं. Now like mitochondria, it was originated due to the engulfment of a prokaryotic cell, and the prokaryotic cell was the cell of cyanobacteria. जैसा कि अपन ने माइटोकॉन्ड्रिया जब पढ़ा था उसमें डिस्कस किया था कि जो माइटोकॉन्ड्रिया है एंड द क्लोरोप्लास्ट, these were originated due to the engulfment of a prokaryotic cells. In case of the mitochondria, there was an engulfment of a alpha proteobacteria, that was Rickettsia prowazaki, while in case of the chloroplast, there was an engulfment of a cyanobacteria. So if you want to study the complete endosymbiotic theory, you can check out my previous video on the mitochondria. So यहाँ सिर्फ इतनी सी बात करते हैं कि जो क्लोरोप्लास्ट है इसका जो ओरिजिन हुआ है, इट वाज ए प्रोकैरियोटिक ओरिजिन, वो एक प्रोकैरियोटिक सेल से हुआ है. So if you talk about the structure of this chloroplast, it have outer membrane which is eukaryotic in origin. We have inner membrane, and this inner membrane, it is prokaryotic in origin. ये किसकी मेम्ब्रेन है? ये बैक्टीरियल मेम्ब्रेन ही है। नाउ द इंटरनल फ्लूइड फिल्ड कैविटी in the chloroplast is known as stroma, which is equivalent to the mitochondrial matrix. Now, the interesting thing over here is that the inner membrane, the inner chloroplast membrane, it invaginates, and after invagination there is a creation of a vesicle like structure, and then these vesicles, they can get detached from the inner chloroplast membrane, comes into the stroma in the form of thylakoids. So these type of thylakoids, these there is a stacks of thylakoids. जिस तरीके से कॉइन एक के ऊपर एक रखे हुए होते हैं, उसी तरीके से थाइलेकॉइड्स के जो इंडिविजुअल यूनिट्स हैं वो एक के ऊपर एक अरेंज होकर के दे फॉर्म्स द स्ट्रक्चर विच इज नोन एज the granum. So we have granum, and granum is the stacks of thylakoid, and thylakoids, these are again are of prokaryotic origin, क्योंकि ये बनी किससे है? These was originated from the inner chloroplast membrane, और inner chloroplast membrane किस कैसा origin रखती है? That is a prokaryotic. तो thylakoid भी क्या है? It is prokaryotic in origin. Now the stacks of these thylakoid, which is known as granum, they links with each other with the help of lamella. Within the stroma we have DNA, since it is prokaryotic in origin, so they have the DNA which is a double stranded circular DNA as found in case of prokaryotes. Similarly, the ribosomes, they are also the 70S type. So this is all about the structure of a chloroplast. Now, if we talk about the function of chloroplast, the major function of these chloroplasts is to carry out the photosynthesis. तो पहला जो important काम है वो क्या है? Is to carry out the photosynthesis. Now, the photosynthesis, it is divided into two parts. One is known as the photo part, other is known as the synthesis part. Now, in the photo part, there is a light reactions. और ये लाइट रिएक्शन किस प्रोसेस से होती है? द प्रोसेस इज नोन एज फोटो phosphorylation, while the other part is known as the dark reaction or the synthesis part, and within this synthesis part we have some dark reactions, and there is a presence of a C3 cycle, we have C4 plant, and we also have some CAM plants. इनके बारे में डिटेल्स में अपन बात करेंगे जब प्लांट फिजियोलॉजी पढ़ेंगे। अभी जस्ट अपन ये समझ रहे हैं कि क्लोरोप्लास्ट के फंक्शंस क्या हैं, तो लाइट रिएक्शंस के या डार्क रिएक्शंस के बजट पे अपन नहीं जाएंगे, जस्ट अपन ये कोशिश कर रहे हैं कि ये क्लोरोप्लास्ट काम क्या करता है। सो द मेजर फंक्शन इज फोटोसिंथेसिस. वी हैव two part of photosynthesis. One is photo part that includes the light reaction, and the process is photophosphorylation. The synthesis part, it includes the dark reaction that have, for that we have C3, C4 and the CAM cycle. And during these dark reaction होता क्या है? जो light reactions हैं ये क्या करती हैं? पहले ये light energy को convert कर देती है into the chemical energy, and that chemical energy is known as ATP and NADH. And during the dark reaction, there is a conversion of a CO2, or we can say there is a reduction of a CO2 into the C6H12O6, that is glucose, by utilizing this particular energy of light reaction. तो light reaction में जो आपने ATP और NADH बनाया है, यही ATP और NADH utilize किया जाएगा for the reduction of CO2 into the ग्लूकोज. सो कंप्लीट रिएक्शन जब अपन बात करते हैं फोटोसिंथेसिस की, वहां अपन पढ़ते हैं देयर इज CO2, देयर इज H2, it will lead to the formation of C6H12O6, there is a generation of water molecule and also the oxygen. So अभी ये बैलेंस नहीं है, जस्ट अपन प्रोसेस डिस्कस कर रहे हैं यहाँ पे. सो ड्यूरिंग दिस photosynthetic reaction we have two processes. There is a reduction of a CO2 into the glucose. At the same time, there is an oxidation of H2O into the oxygen. So we have both the reduction and the you have reduction and the oxidation process simultaneously. So these reactions of photosynthesis, they are called as the redox reaction, having both oxidation and the reduction. तो ये कुछ अपन पढ़ते हैं photosynthesis के अंदर. So जो reduction हो रहा है, that occurs within the dark reaction by utilizing the energy which is generated during the light reaction. Now if we talk about the site of these light reaction, तो यहाँ पे अपन अगर डिस्कस करें तो लाइट रिएक्शंस आपको कहां देखने को मिलेगी? These are on the thylakoid membrane. We can say the site for the light reaction, it is thylakoid membrane. Well, if we talk about the site for dark reaction, then it is the stroma. ये कहां होती है? स्ट्रोमा के अंदर होती है. It is the site for dark reactions. So we have light reaction and the dark reaction. If we talk about the light reaction, क्या प्रोसेस होते हैं? क्योंकि स्ट्रक्चर को अपने को समझना है और इस स्ट्रक्चर को समझने के लिए we have to discuss about the components which are involved in the process of light reaction. So if we take a part of this thylakoid and try to study this portion, then we have four important protein complexes on these membrane which are involved in the process of photophosphorylation. ठीक है, कैसे बात करते हैं? We have chlorophyll A containing photosystem first that absorbs the visible radiation of 700 nm. We have photosystem second which absorbs the visible radiation of 680 nm. Beside this, we have the cytochrome B6F complex and a special type of pump which is known as F-type ATPases. तो चलती कैसे है ये फोटोफॉस्फोराइलेशन रिएक्शन? यहाँ क्या होगा? दो चीजें हैं, एक है फोटो और दूसरा है फॉस्फोराइलेशन. सो लाइट एनर्जी को यूटिलाइज किया जाएगा फॉर द जनरेशन ऑफ ए प्रोटॉन ग्रेडियंट. Then this proton gradient is utilized for the process of phosphorylation to generate the ATP. कैसे बात करते हैं? First of all, the chlorophyll A of photosystem first, it receives the light that is of 700 nm. The chlorophyll A of the reaction center of photosystem first, it moves into its excited state and will leave the electron for another electron acceptor which is the ferredoxin. From ferredoxin, there is a transfer of electron onto the NADH to form the NAD to form the NADH. So there is a production of NADH. जो अपन यहाँ कह रहे हैं, during the light reaction, there is a generation of NADH. Now there is a deficiency of electron within the reaction center of photosystem first. अब ये deficiency कौन दूर करेगा? जैसा अपन photosynthesis में पढ़ रहे हैं, ऑक्सीडेशन किसका हो रहा है? वाटर का हो रहा है. तो इलेक्ट्रॉन डोनर कौन है? वाटर है. इलेक्ट्रॉन एक्सेप्टर कौन है? CO2 रहेगा. या अपन यूं कह दें, जिसका रिडक्शन होता है वो इलेक्ट्रॉन एक्सेप्ट करता है और जो इलेक्ट्रॉन देता है उसका ऑक्सीडेशन होता है. सो जो फाइनल इलेक्ट्रॉन देगा वो H2O है. अभी इलेक्ट्रॉन कहां से रिलीज हुए? फ्रॉम द रिएक्शन सेंटर ऑफ फोटोसिस्टम फर्स्ट. टू मेट द डेफिशिएंसी ऑफ इलेक्ट्रॉन इन द फोटोसिस्टम फर्स्ट, द फोटोसिस्टम सेकंड, इट रिसीव्स द विजिबल रेडिएशन ऑफ 680 nm. The chlorophyll A of reaction center moves into the excited state, leave the electron. The electron will be taken up by the pheophytin. The pheophytin will give their electron to the PQ, that is plastoquinone. It is equivalent to the ubiquinone of oxidative phosphorylation which occurs on the inner mitochondrial membrane. Both of these ubiquinone and the plastoquinones, they are the derived lipids. These are isoprene derivatives. Now after taking the electron, this PQ, it will get reduced into the PQH2, and when it will oxidized into the PQ, it will give their electron to cytochrome B6 and to the cytochrome F. From cytochrome F, there is a movement of electron towards the plastocyanin, and from plastocyanin जो इलेक्ट्रॉन है वो माइग्रेट करेंगे टुवर्ड्स द क्लोरोफिल ए ऑफ फोटोसिस्टम फर्स्ट. तो इस तरीके से जो डेफिशिएंसी यहाँ क्रिएट हुई इलेक्ट्रॉन्स की, वो अपन ने पूरी कर दी कहां से? photosystem second से, but the final electron donor it is the water. So to meet the deficiency of electron at the chlorophyll A of photosystem second, we have a special process which is known as photolysis of water. This is the photolysis of water. That is the lysis of water in the presence of light. ठीक है. So during this क्या होता है, 2H2O molecule है, splitting होगी इनकी किस फॉर्म में, there is production of 4H plus ion, there is a generation of 4 electron and there is a production of oxygen, जो कि अपन यहाँ पे कह रहे हैं during the photosynthesis process there is a production of oxygen. So there is a production of oxygen. Now the 4 electron they will be transported towards the reaction center of photosystem second to meet the deficiency, and at the same time there is a release of 4H plus ion into the thylakoid lumen. ये जो जगह है ये क्या है, ये थाइलेकॉइड की ल्यूमन है, लाइक दिस. यहां पे 4H plus ion आ गए. ठीक है. Now during the reduction and the oxidation of this plastoquinone, there is again a liberation of proton into the thylakoid lumen. Now when these protons they moves back down their concentration gradient through a special type of ATPase which is known as F-type ATPase, there is a conversion of a ADP into the ATP. So we have the energy now, we have the energy in the form of ATP also. So ATP के form में भी आपने energy generate कर ली. So during the light reaction we can say there is a transportation of electrons from one complex to the another complex by utilizing the light energy. The movement of electrons it powers, we can say, the transportation of H plus ions into the thylakoid lumen. Then when these H plus ion they comes back into the stroma through a special type of pump which is known as F-type ATPases, there is a generation of ATP. Now, if we create, or we can say if we compare this photophosphorylation process with the process which is known as oxidative phosphorylation, which occurs on the inner mitochondrial membrane. इस प्रोसेस में अपन ने क्या देखा था कि there is a transfer of electron from NADH or FADH. That the NADH it will give their electron to the complex 1, while the FADH it will give their electron to the complex 2. So there is a transfer of electrons from NADH or FADH to the terminal electron acceptor, that is O2, and the O2 after taking the electron will convert into the H2O. But here what we are observing that there is a transfer of electron from the H2O to the terminal electron acceptor, that is NADH. So we can say we have complete reverse process. ठीक है, वहां से NADH से शुरू हो रहा था, H2 ऑक्सीजन पे खत्म हो रहा था, that is forming the H2O. यहाँ से H2O से start हो रहा है and is ending up onto the NADH. So this process of oxidative phosphorylation, it was we can say along the redox potential, but if you are trying a reverse process then it should be against the redox potential. But this process can be made energetically favorable because we have the presence of light energy over here. Okay? So due to this photophosphorylation, now we have, when these H plus ion we can say when they come back into the stroma through the F-type ATPases, there is a production of a ATP molecule. At the same time, we also have the production of some NADH again towards the stromal side. Then these NADH and these ATP and the NADH they together powers the dark reaction which occurs within the stroma. So this is all about the structure and the function of the chloroplast. Next we will talk about the chloroplast genetic material. The chloroplast genetic material, it is called as the plastome, and since it is originated from a prokaryotic cells, so they have double-stranded circular DNA. The genome size of chloroplast is around 120 to 170 kb. If we compare this with the mitochondrial DNA, and in case of human, the mitochondrial genome size is 16.5 kb, जो yeast में अपने को देखने को मिलती है around 75 kilo base pair. So in both of these cases, we can say that the chloroplast they have larger genome size. Now the chloroplast genome it remains present within the stroma. The stroma contains the chloroplast genome, that is the double-stranded circular DNA. जैसा आपने बात की थी. Now we can say that there is a presence of around 120 genes. These genes they code for the proteins which are mainly involved in the process of photosynthesis. Now, like mitochondria, the chloroplast is again a semi-autonomous cell organelles, means since they have their own DNA, but they are not able to synthesize all of their proteins. So, they also depends upon nucleus for their some of the proteins. And to understand the semi-autonomy of chloroplast, we have classical example of RUBISCO. Now, RUBISCO, it is an important enzyme of dark reaction. The most abundant protein on the earth crust, it is the RUBISCO. The most abundant protein in human body, it is collagen. The most abundant protein in human blood, it is albumin. And the most abundant protein in human RBC, it is the hemoglobin. ये चीज भी अपने को याद रखनी है. So, RUBISCO, it is a most abundant protein on the earth crust, important enzyme of dark reaction. It is made up of 8 large and 8 small subunits. Now, the 8 large subunits, these are the catalytical units which are encoded by the ctDNA, that is chloroplast DNA or the plastome, while the 8 smaller subunits, these are encoded by the nucleus or the nuclear genome. Within the nucleus, as we all know that we have the DNA, there is a process of transcription. This transcription leads to the formation of a mRNA. The mRNA, it comes out into the cytoplasm. Within the cytoplasm, there is a binding of ribosome onto it, lead to the production of proteins through a process which is known as translation. So due to the translation, we have the protein within the cell cytoplasm. As we all know that like the nucleus, the chloroplast, they also have the cytosolic protein. Some of the protein of these chloroplasts, they are cytosolic. So these proteins, they will be targeted into the chloroplast. Now we have protein within the chloroplast. Suppose this protein is for the 8 smaller subunits of the RUBISCO. Similarly, the chloroplast, they have their own DNA, forms the mRNA, and this mRNA, it forms the protein. And suppose this protein, it codes for the 8 larger subunit of the RUBISCO. Now, these 8 small and 8 large subunits, they together forms a functional RUBISCO enzyme, which is the important enzyme of dark reactions. So in this way, we can say that even for the complete, we can say functional RUBISCO, this chloroplast, it depends upon the nucleus for their smaller subunits. So we can say the chloroplast, it depends upon nucleus for their some of the proteins. That's why it is a semi-autonomous cell organelle. Next is the maternal inheritance. Like the mitochondria, this chloroplast, they also shows the cytoplasmic inheritance. Means these mitochondria and the chloroplast, these are the part of the cell cytoplasm. और cytoplasm is generally donated from the maternal side or the mother side, इसलिए इसको अपन कह रहे हैं cytoplasmic inheritance, क्योंकि ये एक पेरेंट्स की तरफ से ही डोनेट हो रहा है. That's why it is also known as the uniparental inheritance. Now the classical example to understand this maternal inheritance in case of plant is the variegated leaves of Mirabilis jalapa. It was observed that in case of Mirabilis jalapa, there is a presence of 3 types of plant. The plant which are having the green leaves, we can say. We have the plant with the white leaves, So we have white leaves plant, and we also have a plant having the mixture or having the patches of white and green, which is known as variegated plant. Now, this variegation, or we can say these leaves pattern in case of the Mirabilis jalapa, these are due to the plastids. ये generally plastids की वजह से ही है क्योंकि pigments they generally remain present within the plastid. So the plastids are responsible for these different colors of leaves in the Mirabilis jalapa. तो इसको और स्टडी किया गया. When we have taken a white-leaved Mirabilis jalapa as a female plant, ठीक है, that is the egg cell. Regardless of the pollen grain, they always gives the white plant because we can say that it is a cytoplasmic inheritance. प्लास्टिड किस तरफ से आएगा? मैटरनल साइड से आएगा, और मदर का जो फिनोटाइप है वही किस में आएगा? प्रोजेनिस के अंदर आएगा. सपोज इफ वी हैव टेकन ए green plant, regardless of the pollen grain, it will again give the green progeny क्योंकि मैटरनल फिनोटाइप क्या है? ग्रीन है. Similarly, if we take a variegated plants as a female, then it will gives all variegated progenies. Because these types of leaves or the leaf patterns, they are they are influenced by the plastid, and the plastid it have the maternal inheritance, that is known as the uniparental inheritance. So these plastids, like mitochondria, they shows the cytoplasmic or the maternal inheritance. So this is all about the chloroplast. Thank you very much.